Welcome to the Chapter 10, Section 3 podcast, Regulating the Cell Cycle. And we're going to be talking about how the cell cycle is stopped, started, paused, etc. So let's start. We're looking at a picture of a scar there because we're going to be talking about cell division and how it helps in healing. So you have to control cell division here. And obviously when you have a cut, you need to replace that missing tissue, that missing skin with more cells. So you're going to have a lot of cell division at that site. And when you're done with that, you're going to need that rapid division to stop, and it needs to slow down and then return back to normal. So what they've found is that normal cells will reproduce until they come into contact with other cells. And when they come into other cells, they don't grow anymore. And what this is showing is that cell growth and division can be turned on and off, just like with the scar. So let's look at this example here. We have a petri dish with some cells in it, and the cells are allowed to grow. And if allowed to grow, they'll grow completely to the edge of the dish and crowd with each other, and then they will stop. Now what we'll do is we'll take some kind of tool and scrape out a line of cells in the middle of the petri dish. And what we'll see is that the cells will start to grow back to replace that space, and then once they come into contact with each other again, then the cell growth stops, and we call this contact inhibition. So how is the cell cycle regulated? It's regulated by a specific protein, and the protein rises and falls in time with the cell cycle, and they call this protein cyclin because it regulates the cell cycle. And if you look at this picture right here, you can see there's a couple different types of cyclin, and they each come into play at different points during the cell cycle, triggering the different parts of the cycle. So cyclins regulate the timing of the cell cycle in eukaryotic cells. So cyclins were discovered during an experiment, and what they did was they took some cytoplasm out of a cell in mitosis, and then they ejected that sample into a cell that was in the G2 of interphase, and what had happened, as a result, it triggered that cell to enter mitosis, which was interesting. It took a cell that wasn't in mitosis and forced it into mitosis. Now we have two different types. We're going to separate regulators into two different types. Proteins that respond to events inside the cell are called internal regulators, and they allow the cell cycle to proceed only when certain processes have happened inside the cell. For instance, like the production of the right amount of organelles that the cell will need after division, or when the chromosomes have been duplicated during cell division. External regulators respond to events outside the cell, and what this will do is it will direct cells to speed up or slow down the cell cycle. And a couple examples are embryonic development and with wounds, that we'll have regulators outside the cell. For instance, like we saw with the cells that were with contact inhibition, once they touched up against another cell, they would slow down the cell cycle so that they wouldn't divide anymore. Now, uncontrolled cell growth, and this is another topic. We're talking about cancer here, and what happens is with a cancer cell, it doesn't have the ability to control its growth, and they don't respond to the signals that regulate the growth of most cells. And what they'll do is they'll divide uncontrollably, and they form tumors, which are just clumps of cells, and then these tumors can then damage the surrounding tissues. They can squeeze out other tissues. And what could also happen is that if the cells break loose from the tumors, they can spread throughout the body, and thus you can end up with cancer in another part of the body. Now the quiz.